Okay, so we have been busy. So you saw an old 2014 lecture on an introduction to Mendelian inheritance. And I think it works okay. I mean, it's a little fuzzy. Now we're going to kind of continue where we left off. As a matter of fact, I'm going to put that last example from that old lecture on the board, and then we're going to roll on into beautiful complexity for Mendelian inheritance. So as you remember, we had this cross that we were doing at the very beginning where we had identified two different parents. So I guess I would put that these are our parents over here. Remember that that each of these letters is an allele, and that this indicates therefore a genotype. So this is a genotype here, which we are abbreviating as GT. This is a genotype here for the other parent. The phenotype for this particular parent was purple flower, and the phenotype for this plant was white flower. And so this is classic Mendelian inheritance here. And then what we always want to do, I always like to throw this in a cross. I always like to indicate what the gametes are going to be. And when I put a gamete there, I'm going to put one allele, because there's only one instead of two, because it's haploid, right? And I'm going to circle it. So this parent, all of the gametes are going to be big P's. This particular parent, all of the gametes are going to be little p's. And then, basically, therefore, this represents kind of what we would call an inbreeding cross. Or excuse me, not this. I got ahead of myself. This represents this parental cross. So all of the offspring are going to be big P, little p in their genotype, and they're going to be purple flower. So therefore, that's the genotype, and then they're all going to have purple flowers. And that is the phenotype. Then, now we get to do an inbreeding cross. So we want to do an inbreed. Now, of course, think way back to the beginning of that lecture when we were breaking down a pea plant and showing the parts and showing how Mendel could control pollination with basically a little paintbrush and some snipping and some bags. Yeah, that kind of a technique. So we're going to breed these two now. So we've planted the seeds, grown them up, they've produced purple flowers. And then we're going to cross one of these purple flowering plants with another purple flowering plant from this same cohort here. And now, therefore, our cross is going to be big P, little p, big P, little p. And I'm going to erase the top of the board so we can follow this up at the top. So, now, therefore, up to the top, big P, little p, big P, little p. This is our cross. Now, for gametes for this particular cross, this is going to be a 50-50 thing. Meiosis is going to produce half of the chromosomes with the big P and half of the with the little p. So therefore, we have big P, little p, and then this parent, big P, little p. Now, as we saw in that previous example, what we're going to do is simplify our predictions for this cross. We have 50% these, 50% these, and then for this parent, 50%, 50%. We're going to basically create this Punnett square, the Punnett square, which is this great tool to predict the way that all these possible gamete combinations can form. So big P, little p, big P, little p. And then we basically we bring column and row together to produce the possible genotypes of offspring. Big P, big P, big P, little p here. And you always, by the way, if you haven't noticed, you always put in a heterozygote, you always put the dominant allele first, or the same allele first, if we get into other allele situations in a minute. And then we have it. Now, here's where we need to introduce very important new concept. What we want to do first off is tally the find out what they are. So I'm going to use purple. It doesn't show very beautifully as purple, but I think you can see that it is different. All of these are purple offspring. And then this guy here is the only one that's white. So let's go ahead and break all this down into two different kind of assessments. The first one is going to be what we call the genotypic ratio. And that's something you should get used to calculating. And in this case, we're going to go 1 to 2 to 1. So we have a 1 to 2 to 1. We have 1 homozygous dominant, 2 heterozygotes, and then 1 homozygote recessive. Then the next thing for this cross is our phenotypic ratio. And this ratio is this. Those are the purple ones, so it is 3 to 1. And of course that would be purple to white. That's our phenotypic ratio. You should always be prepared whenever you do a cross to actually list genotypic ratios and phenotypic ratios. Matter of fact, that's one of the keys to solving the various problems that you have in a problem set of typical inheritance problems. We'll look at a lot of those. I've got a good set. We'll be kind of going through those in our optional meeting. And you'll be doing a lot of them on your own for credit in terms of your punch cards and just preparing for the final exam. So there we have it. Now we're kind of caught up. This is a summary of what we did last time. It's now time for us to dig deeper into the concept of variations that can occur when you do this, what's called monohybrid inheritance. I want to let you know one other thing. What we just did classically would have been called a monohybrid cross. You're going to see this language. Mono means one, hybrid means heterozygote. And so here we're basically looking at one trait and it's heterozygous at that trait, and in a cross we're crossing two that are heterozygotes. And you should always know in a monohybrid cross with normal dominance recessiveness that you're going to get a 1 to 2 to 1 genotypic ratio and a 3 to 1 phenotypic ratio. After I go through some kind of just reorganizing of our terms, we're going to get back into what monohybrid crosses do when the rules are broken. Because there are variations, non-Mendelian variations, that have to do with the ways that alleles express themselves. That's coming up. Okay, so let's get busy here. Let's get busy here with just kind of a summary of some of our terms then. So you've seen these terms before, but it's good to write them down again and get this all tight. So we already know we've defined a gene, we already have defined a locus, we've already defined alleles. And this is the one we really want to expand and get new stuff. I'm going to bring some really cool ideas out. But remember that a gene is a specific, I'm abbreviating, sequence of bases. And this codes for the specific amino acid sequence. And then a locus is the location of a gene on a chromosome. Abbreviating these, you already have these definitions, sometimes a couple of times over. Alleles, alternate forms of genes. Now, what we've done with this is we've kind of expanded this. Here's some new stuff then. Here's new to us. We have dominant alleles, and let's see here. We have dominant alleles. These express their phenotype when present. we have what we call recessive alleles. And these express their phenotype only when homozygous. Only when homozygous. And continuing down our list, some really new ones, some good new ones, and I'll bring these examples up in a minute. Codominant alleles. This is cool stuff. Now, codominant alleles, both phenotypes are expressed in heterozygotes. So you're going to see examples of that in a minute. And then the next one is incomplete dominant alleles. And in incomplete dominant alleles, the heterozygote produces an intermediate phenotype. Okay. So that's really good stuff, kind of to set us off. One thing I want to add, with a little bit of a break from these types of alleles, is a new situation called multiple alleles. And we save this concept for having greater than 2 types of alleles at a given locus. Okay, there is a beautiful set of concepts. We have already completely knocked this one out. We saw in our example, we saw this with the purple flower, white flower cross system that we looked at. So we've seen that if a genotype had a big P, it was purple. And we saw that the only way to be white was to be, to express the phenotype only when homozygous. So we've seen that with that first example from today and from the end of the previous lecture. So now what we want to do is pick up on these examples here of codominant alleles, incomplete dominant alleles, and multiple alleles. I'm actually going to start with incomplete dominant alleles. So what we're going to do now is a cross, and one of the classic crosses that we'll see is coloration. Let's see. Got to find my genotypes here. I want to be consistent with the problem set. Let's see. All right, so here we're going to go. We're going to look at R. This is cattle. R is an allele that goes for red coat color. And big R prime is going to be for black, I believe. Let's double check this. Come on now. Let's see. Just black coat color. And actually, it's brown. Brown coat color. And then what we're going to see in the genotypes for incomplete dominance is that this guy here is red. This guy here, that's the heterozygote. That one is roan. And that's the big point. Incomplete dominance is that the heterozygous phenotype is intermediate. Roan is a color between red and brown. So then we have this guy here. That's the only way to be brown. So this is an example of incomplete dominance. Notice there's a little code. Notice for allele assignments that I'm using caps, cap and cap prime. The capital letter indicates I'm bringing a punch. My phenotype's coming in. And in this particular case, it's an intermediate phenotype that's produced. So that's incomplete dominance. The next one, we're going to put two things together. We are going to do two things at once. We're going to do codominance, and multiple alleles in one system. So our example for this, so in codominance, if you look at what I defined, I said that that the phenotype of each individual codominant allele is expressed. So our example for both of these things is going to be human blood type. So here's the alleles. We have I super A, I super B, and these are codominant. And then we have little I. So therefore, in this system, here's the possible genotypes. The possible GTs, and we have I sub A, and I'm actually going to put in comma I sub A and little I, because both of these have the following phenotypes. This is type A. Okay, let's see. Because I'm creating kind of a table here. And then I have I prime B, and then I prime B and little I. That's the heterozygote for type B. And then we have the special one, which really defines the codominance, right here. This type is type AB. That's the one we want to really... That's codominance. Codominance. Each one of these alleles is expressing its phenotype. Therefore, the phenotype is called AB. And then we end up with the last possible, which is the homozygous recessive, and that's what we call type O. So there you have, in one frame, a beautiful situation that describes multiple alleles. I'll go ahead and put circles there. So here we have 3 in this example, and we defined multiple alleles as being at a given locus greater than 2. So in our genetics problem sets, we'll be working some nice blood type kind of paternity problems, where you always know who the mother is, right? Because it's the pregnancy thing. But who's the father? Paternity cases and blood type, those are fun. Variations in monohybrid crosses. So, variations in monohybrid inheritance. You're going to take a look at some of these. I've already kind of given you this. What we want to introduce is kind of the concept of ratios. So we want to think about ratios. So this first one would be would be what we call complete dominance. So in complete dominance, a monohybrid cross yields. So a monohybrid cross yields the following. And by the way, by monohybrid cross, I mean big A, little a, cross big A, little a, or variations therein. I super A, I super B, cross with I super A, I super B, for a codominant cross between type ABs, for instance. So a complete dominant situation would be 1 to 2 to 1 genotypic ratio and 3 to 1 phenotypic ratio. And then the next kind of cross that we can mess with is incomplete dominance. So in an incomplete dominance monohybrid cross, you would get a 1 to 2 to 1 GTR and a 1 to 2 to 1 PTR. We'll work some problems. You'll see some problems that show that, but I think that it's good to put your put your brain in contact with ratios. The next one is a codominant cross. Codominant, and again, this would be a 1 to 2 to 1 to a 1 to 2 to 1. Let's do an example of this just to see. I'm just going to insert an example. So we got type AB male reproduces with a type AB female, and that's what we're going to have then. So let's set up the cross. The cross is I super A, I super B, crossed with I super A, I super B. So these are the parents of the cross. The gametes of the cross would be 50-50. Half of them from this parent being A, half of them from this parent being B. Cross with the same. Cross with the same. So then we have that. Everybody knows here the next step is 2x2 punnet. So again, I'm going to work the top of the board here just to kind of give the space that we need. So here we have this cross. Didn't leave myself a lot of room. Boy, got too excited there. That's the cross that we just set up. So now I just wanted to show you, here's type A. Here's an individual that is AB, like the parents. Here's an individual that is AB, like the parents. Here's an individual that is B. So as you can clearly see, we've got one type A, and we've got two. So let's get a 1 in front of that. 1, 2, 2, which would be AB. I'm just going to change that to 1. Actually, I'm doing a genotypic ratio. So I was doing it right. 1, IA, 2, 2, IA to IB, to 1, IB, IB. So it's a 1 to 2 ratio, 1 to 2 to 1 ratio, and then the phenotypic ratio. So this is our GTR. Our PTR for this cross would be the same. It would be 1A to 2 ABs to 1B. That's our phenotypic ratio. 1 to 2 to 1, 1 to 2 to 1. It's the same. So just wanted to insert that there so you could see that. It's really good to get your brain wrapped around phenotypic ratios. Okay? Good stuff. Hammering on down. Now, the next part of our fabulous story is to introduce an interesting concept. So we've got multiple alleles under our belt. There's another concept here we call pleiotropy. And pleiotropy means one genotype yields greater than one phenotype. And our classic example of this is going to be sickle cell disease in humans. Now, we're going to tie this into some of our great early work. We're going way back to proteins. And you'll remember that hemoglobin, the oxygen, the gas transfer pigment in our system, is 2 subunits. It's a quaternary protein, so it's 2 alphas and 2 betas and then heme groups, right? So this hemoglobin molecule is a protein, and therefore it's a sequence of amino acids. That's the primary structure, and then the secondary structure established by hydrogen bonding, alpha helices, beta pleated sheets, tertiary structure, even more intimate, twisting and turning and shape building, and of course then the quaternary structure. So very complicated and beautiful protein. We have many of these proteins in our system. I've already talked about them once before, just a tiny bit in that previous lecture. Now we're going to think about what could happen to that if we had one point mutation. A point mutation in DNA that changes the amino acid at a particular location, which changes the structure altogether. So here's what we have in sickle cell disease. This is a 2-allele system. So it's a standard 2-allele system. And we have one, which is HBN. This is a slight variation from the general practice of naming alleles, of symbolizing alleles. We usually use one letter. And then, of course, if it's codominant with a prime, more on that later. So we have HBN and we have HBS. So this one, as you might guess, is normal. This one, as you might guess, is going to be the sickle cell mutation. So, in this particular case, let's draw the possible phenotypes, or excuse me, the possible GTs and kind of the PTs. We would have here a HBN, HBN, and that produces, from a circulatory perspective, this is the genotype, and then the phenotypes, this would be normal, and by the way, this is phenotype number 1, normal kind of O2 transport. And this is the curveball, susceptible to malaria. Just let me write this on the board before I explain it. So here we have, matter of fact, I'm going to modify this table right here. We just run out of space sometimes, so I just need to do this. I'm going to make it stand out. I'm making a table here that is going to have possible genotypes and then associated phenotypes. And we have 2 different phenotypes. So this is associated PTs, and note that I have 2. I have basically O2 transport, which means that if you've got nice, beautiful cells, they go through and do the job. If you have cells that are sickle-shaped, then they have problems. And I'll explain that in a minute. So O2 transport, and then over here, this would be regarding malaria susceptibility. So I guess what I've done here is one genotype yields greater than one phenotype. So here's this concept here. 1, 2 phenotypes here in this particular example. So notice I'm following each phenotype. Here is HBN, HBS. This person, O2-wise, has sickle cell disease, and this indicates kind of impaired O2 transport. Now, the basis of the impairment is that these cells here look different. When you are normal, your red blood cells are just perfect little BBs. That's a red blood cell. And when you are HB and HBS, you end up with half of your cells being this, and then half of your cells being kind of this. And so this guy right here, that's the problem. That's a sickle cell. And I'll explain this here in a minute. Here's the deal. Explain it now. So all of the cells that have what's happening is we're producing proteins from both, and this gene codes for cells that end up like this, and then this one for the normal ones. So some of your cells, if you have sickle cell disease, when they're going through your capillaries, and the idea of a capillary is it's the smallest unit of your highly branching circulatory system. You know, we've got big aortas and things like that, and then it gets smaller, and we have arteries, and then we eventually get into the tissues where it's so finely branched they're into capillaries. And capillaries are just barely big enough to be the barrel that holds red blood cells. So these red blood cells are kind of, flying through these barrels, and it's very efficient. But if you've got a clanker that's shaped like this instead of like this, it's like bing, boom, 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 bing, bing, boom, as it gets through the tiny little capillaries. And then every once in a while you get roadblocks, and they just stop. And of course, if that's happening, then your tissues are being deprived of the oxygen that they need. And then of course we go right back to our aerobic respiration phenomenon, and we looked at the examples of what happens when it goes anaerobic. Do you guys remember lactic acid? Yeah. So that's happening right there if you have a roadblock of sickle cells. These oftentimes self-resolve. Sometimes they take drastic measures. Sometimes you kind of have to, you whap it, It's pretty crazy stuff. So here we have an impaired, circulatory impaired person. However, the flip side of this is that these are resistant to malaria. So the malaria parasite is an intracellular blood parasite that attacks red blood cells and gets inside and then starts to asexually reproduce to make more copies of the parasite, and then it ruptures red blood cells and invades another. It loves beautiful, round, normal red blood cells, but it does not do well in sickle cells. So here we have an interesting thing. This is good in a world of no malaria. This is good in a world with malaria, because this is bad. So this is bad. And then finally, let's just give service to this last one, HBS. This is a person that is quite sick. I guess that what we would say here is that malaria doesn't even really matter to this person, because this person usually dies fairly early in childhood. So most die in childhood. So there we have this concept of pleiotropy, very important, and a curveball for Mendelian inheritance. So here you have a place where one genotype, and here I've listed 3, but for every 3 in this concept, I have identified 2 different phenotypes. So especially in Africa, this is where this is the most abundant, because there's endemic malaria and there's sickle cell disease, and you could see that you would think, if you start to think about, evolution in populations, this is something that happens much later on in your biological progressions as you get into population evolution and ecology and genetics and all those good things, you would see this little tagline here. You would see that in areas where both malaria is present, or where malaria is present, that these guys would have a disadvantage. These guys always have a disadvantage. These guys would have a subtle advantage because malaria is not killing them. But of course, these guys are always going to have an advantage over these guys because, in terms of oxygen transport, because they're not sickly. They have normal, high-functioning circulatory systems. So you have this situation where, because of malaria, this HBS allele does not just disappear from society because of its bad effects on your circulatory system, because it has counter effects in terms of malaria, which takes a lot of people out in those parts of the world. Exciting stuff. Okay, so now I think we have covered a wide variety of exciting kind of variations on classic Mendelian inheritance focused on just one locus. So one locus with two alleles, that's our bread and butter. That's where we started. We got a dominant, we got a recessive, we've got homozygous dominant possibilities, heterozygous, and homozygous recessive possibilities, but then there's curveballs. The last thing we need to do before we leave this topic of monohybrid inheritance is introduce one nice tool. So, there is something we need to introduce that's called a test cross. So a test cross is that an individual with a dominant phenotype and, of course, unknown genotype. And do you know how I would represent this, by the way? I would represent this as, let's say that it's in the flower system. For example, the way that I would say this is P question mark. And that is a purple flowering P, but it has an unknown genotype. So that is P question mark. So in other words, in a test cross, what you would do is take this this individual with a dominant phenotype and an unknown genotype, and cross it with a homozygous recessive. Now, of course, the beauty of the homozygous recessive is that you know its genotype. So therefore, here's what the test cross is. P question mark with little p. This is a white, this is a purple, and the test cross outcomes are simple. If question mark equals P, big P, then they're all purple. If in the cross, if in the cross there's a 1-to-1 ratio of P, of purple and white, it is a heterozygous, big P, little p. So let me see. Question mark is equal to little p. And so therefore it's a heterozygote. So that's a test cross. And that's a good way to get what's called a 50-50 ratio. So I guess I could write here that in this particular case, when it's this, your GTR is 1 big P to 1 big P, little p, and your PTR is 1 perp to 1 white. There you have it. So there's another ratio for us to get the test cross ratio when you have an unknown dominant phenotype, meaning of unknown genotype. crossed with, in the test cross, it produces this. Of course, if you wanted to know what this ratio is, the GTR is 1, the PTR is 1. That's not really a ratio because it's all, it's one big P, and it's one big P, little p. Or excuse me, the phenotype, it's a one purple. That's all they are if question mark is big P. Okay, there we have it. That's pretty good stuff. I think we are ready to advance into the wonderful world of what happens if we start to add some new loci to the system. Okay. Alright, now it's time to get funky. This is going to be a lot of fun. I need to go back and we've got to take a look at meiosis. We've got to take a look at kind of what's going on in meiosis. What I want to do is introduce this concept of we are now going to basically follow greater than 1 locus. Now, we've got to get some locus, kind of loci concepts back behind our belt. Let's take a look. Let's go back to late prophase 1 in meiosis, of course, and take a look at a situation where I'm going to have, we're going to look at 2 chromosomes. So I'm going to have 2n, n equals 2. And of course, we're going to use the classic blue and red to get this story done. And then I'm going to identify some things. So 2n, n equals 2. And of course, that means we'll have a long chromosome and we'll have a short chromosome. So the whole cell's looking like this at this point. Nuclear envelope is essentially gone. Of course, just to give honor to what's happening, it's almost ready for metaphase, so we've got a beautiful spindle forming. What I really just want to do, though, is show you that, hey, right here, by the way, I'm not going to show any crossing over. It's going to get us confused. What I'm going to do is show you that we've got a big duplicated chromosome here, and we've got a little duplicated maternal here, and we have a big paternal and a little paternal. Whoops, that should be a little bit bigger to make it the same size as the one that I drew over here. So there we have our long maternal paternal, and these of course are duplicated chromosomes, 2 chromatids in a centromere. That's what we got, and that's what happens in prophase 1. Just to let you know one more time, crossing over would have already happened, but I'm not messing with that here. We're going to imagine that it didn't happen for these 2 chromosomes that we're following. So what I want to do is say that we have locus A, we have locus B in green, and let's get one more. Let's do, that looks kind of orange and probably won't stand out much. Let's see if I can find, let's try yellow. God, that looks kind of like orange too, but I'm going to try it. Locus C. What I'm going to produce for you here is, I'm just going to write it in white. I'm going to produce big A, big B, big C, little c. So we're actually following, in this case, 3 loci. And I'm going to show you that we have locus A is living right here on the big chromosome. And I'm going to show you that that's a big A, that's a big A, that's a little a, that's a little a. It has to be that way because it's a heterozygote. And of course, these are 2 chromatids. They're not chromosomes yet, so the reason that there's 2 on this one is that each chromatid, this was product of DNA duplication. Then we go to B. We're going to say that B lives on the same chromosome, and We'll call this one, we'll say that maternal was the dominant and the paternal the recessive. And then over here on, I'm going to have, whoops, this should be little c and little c. And then over here, big C and big C. So basically I've attributed all of these, in this particular case, to the maternal, and then all of the recessives to the other. I just want to get everybody in the same boat here. These are linked loci. Linked means they're on the same chromosome, which I'm abbreviating. And then A and C, as well as B and C, these are what we call independent, which I'm abbreviating, loci. So this means that independent loci are on separate chromosomes. Or on different chromosomes. Now, we will focus our attentions on independent loci. So our big party is right here. We are going to spend our time dealing with independent loci, at least for the next good chunk of this presentation. It's exciting. We can follow two things at once, and it really begins to actually make us better meiosis people, because we have to think back to meiosis and independent assortment to come up with gametes. So, and by the way, you just heard that word independent assortment, or that concept. Independent assortment, independent is huge. Separate chromosomes. All right. So let's take a look at an exciting Here we go. We are going to do a dihybrid cross. Of independent loci. With independent loci and complete complete dominance at each. Our system, we'll go to peas, and we're not going to be looking at flowers, we're going to be looking at seeds this time. So we are going to have big Y, which is going to be yellow. Little y is going to be, this is a pea seed. Little y is going to be green. So this is dominant, this is recessive. And then we're going to also look at R, which is going to be a round pea, and a round seed, that is. And then little r is going to be a wrinkled one. A wrinkled seed. So there we have it. So we'll set up our cross. I'm going to get this down now because I'm going to erase the board because we need a lot of real estate. Okay. So here we go. We're going to do, therefore, in our parental cross, we're going to do, we're going to make dihybrids. And to do that, we're going to go boom, boom, boom. So this is basically what we would call true breeding or homozygous, yellow round, with true breeding or homozygous green wrinkled. So that's what we're going to do. So this is, I'm going to say, true breeding. That's the old Mendel term. True breeding is a synonym for homozygous. So homozygous yellow round, and we're going to cross those with true breeding, which is heterozygous. Green wrinkled. Are you with me? Okay. One last thing to say about this, again, is that true breeding is an old Mendelian concept, and you'll often see that word in genetics problems. Basically, instead of homozygous, they would use true breeding. So be used to that. You should be able to use this complete terminology interchangeably. Then the heterozygous synonym for Mendel is hybrid. And whenever that appears, you should be thinking heterozygous. So there we go. Now, what we're going to do then with this cross is follow down and identify their gametes. All of the gametes here, this is the new part. When you follow more than one locus, the gamete has to have an allele for each locus. So this is 2 loci, we need 2 letters. Big Y, big R. They can only get a Y, a big Y, it can only get a big R. They're on separate chromosomes. Big Y, big R. These guys, of course, little y and little y and little r. And then, of course, this is our parental cross. So what we're going to get in our offspring, these would be what we call F1s, and I don't think I've covered this. This is our first filial generation, this is what it's called. F1 is our first generation from this controlled cross. All of these offspring are going to be, quite obviously, big Y, big R, little r. In case you wanted to know what that would look like, that's all you got. You can only make, if that's the male gamete, that's the female gamete, and that's all you can get from that. Now, that's their genotype. All of these, phenotypically, all of them are, because of normal dominance and complete dominance, they are all yellow and round. Okay. Now what we're going to do is we're going to grow them up. We're going to grow up, get seeds, grow them up and get flowers, or basically do our controlled pollination here. And this is going to be what's going to be inbreeding. We're going to inbreed these guys. We're going to breed them together. So, erasing the board again, we're going to set that up. Okay, so now let's set up where we left off. We're going to do a cross here of big Y, big R, little r, crossed with big Y, big R, little y. And by the way, this is the dihybrid cross. And here is the crazy cool part. Let's do gametes here. So here's our possible gametes. Now, you're going to have every combination of these. I'm just going to lay them out. You're going to have this gamete from independent assortment. And if you need help, go back to meiosis. Go back to that exercise that we did with coin tossing and independent events. Basically, that's a coin with a head and a tail. Big Y, little y, head, tail. That's another coin with a head and a tail. So we have 2 heads. Then we have a head and a tail. And then we have a tail and a head. And then we have a tail and a tail. Or you could figure out how many gametes with your x to the n. X being the number of outcomes per event, n equals the number of events. The events are 2. 2 squared equals 4. We've got 4. Are you with me? That's that parent. Sliding this one over here, exactly the same. Big Y, big R, head, head. Big Y, little r, head, tail. little y, big R, tail head, and finally little y, little r. Always, when you get into these complicated crosses, always keep your gametes in the same order. We're going to therefore turn these gametes into our rows. We're going to turn these gametes into our columns. We've got 4 and 4, so we need a 4 by 4. So let's put them in. So on this side, big Y, big R, big Y, little r, little y, big R, little y, little r. Same order, down as across. And then what you do is match them up. Big Y, big Y, big R, big R. Guys, just start going. Just go to town. Big Y, big Y. Just go to town. I'm going to stop saying things because this is pretty complex. You're actually going to see a nice trend, however. If you have everything lined up, I already know that I can just go ahead and fill this thing in because it's going to be dihybrids all the way down the all the way down the... I did dyslexia here. I don't know if you saw that. Big Y, little y, big R, little r, big Y, little r, or little y. There we go. Sorry about that. Okay. And then just fill in your others. Whoops, I'm running out of space in here. Yay! There we go. All the possible offspring. And what we're going to do next is play with the concept of the ratios. I've got to take a break now because we have a meeting. It's approaching 11 o'clock, if you can't see. And then we'll come back. I won't touch this board. We'll come back and we will make genotypic ratios and phenotypic ratios. And man, that's when it gets fun. Phenotypic ratios of a dihybrid cross. It's a really cool thing. Okay, so see you soon. All right, so now we got some work to do with this list. This is fantastic. I'm going to erase this little arrow. What I'm going to do is I'm going to start to do a tally on this in the column. So the first one is we're going to make a total list of all the genotypes. I'm going to start up a little higher. Here's the genotypes. The genotypes that we get from this, and I'm going to make a tally list, and then after that I'm going to put the phenotypes. So this is going to be a little table that goes with these results. So first off, I'll do that and then put a check mark, and I'll do it with a color so we can see it. So here's Y, big Y, big Y, big R, check. Just continue down the line and write the new ones as you get them. And so, guess what? I already did that one. So, there was one of those. There's two of those. Apologize for this. There's one of these so far, and there's one of these so far. Okay, now, big Y, little r, got one of those, check mark. Then we have another one of these, big Y, big R, check mark. That lives here. So now we're doing tallies. Big Y, little little r, first one of those. Check mark. I think I've got this guy already. He lives right here. Check mark tally. This guy here, of course, we have. Check mark tally. Then little y and big R's. No, this is new. Little y, big R, big R. Tally check. Whoop, tally check mark. Yes. And this guy we do not have yet. It's our first little y one. Our second, actually, little y. Big R, little r. Tally. Oh boy, I'm running out of space. Got another one of these. Check mark tally. This guy I've got. Check mark tally. And this guy I've got. Check mark tally. And then finally, little y, little r, and check mark tally. Oh my God, we pulled it off. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 different genotypes. So that is crazy. If you wanted to write the genotypic ratio, the GTR, it doesn't even make a lot of sense. Basically, the way that they usually present it is it's a 1-to-1 to 2, 1-to-1 to 2 to 2 to 4 to 2 to 2 to 1 to 1. Like, that doesn't give you any information at all. Genotypic ratios are really valuable when we're dealing with one locus and we're doing those simple things. That's not really a very good kind of why is the question. But the phenotypic ratio is much better. So here is what we've got. We have all of these, my friends, are going to have dominant for the phenotype because they have at least one capital letter allele for each locus. 4, 5, 6, 7, 8, 9. There's a total of 9 of these. And we know there's a total of 16, so 9 out of 16 of these are going to be yellow round. Yellow round, 9 out of 16. These ones all line up beautifully together. 3 out of 16 are going to be yellow wrinkled. And then this group here, 3 out of 16 is going to end up being green and round. And then this little guy here, 1 out of 16, is going to be green, and wrinkled. Boom. So phenotypic ratio is big time stuff. It means something. 9 to 3 to 3 to 1. This is very, very important for us because we are going to start to manipulate our crosses by doing different kinds of alleles. And we're going to see modifications of the dihybrid cross of independent loci. We're going to see modifications in the phenotypic ratio of that of different kinds of dihybrid crosses. This is the big one though. Very, very cool. Now, I want to show you a little cheating event. So, I'm going to erase this carefully. And I'm going to keep all of my nuts and bolts down here from the genotype-phenotype stuff. I want to ask a question here using a different system. So we know that each of these things is an event. So we have a monohybrid cross. I'll actually change the colors. So we have 2 events. These are independent events. We have This is in the red. I guess I just want to write somewhere down here. These are each locus is a different independent event. So what we could do is start to work problems like this. For instance, I could ask the question without looking at this. Said, how or what proportion of offspring in this cross would be big Y, big Y, big R, little r? Question mark. I want to know what that is. We have the answer over here, but that's what the big, huge 4x4. This is the shorthand. So what you need to do to answer this is bring this guy down and do a monohybrid here. So you know how many of these are going to be that? 1/4 are going to be big Y, big Y. And then over here, it's going to be the same deal. Big R, little r, big R, little r. And what we're looking for would be these heterozygotes. So we'll have 1/2 of these individuals will be big R, little r. Then it's basically 1/4 times 1/2 equals 1/8, which actually would be 2 out of 16. Look at that. So the one we're looking for right here is this guy, 2 out of 16. Ha ha! That's the shorthand. That's an awesome shorthand. Now I'm going to present it one other way. So I'm going to erase this, get a good look at what I've done. We'll do more of these later, especially in our little worksheets, our problem sets. Okay, so we're saving all the data from the full 4x4 Punnett square. We did a 4x4 to do this complicated cross. It was a dihybrid cross of independent loci. It required a 4x4. Let's ask a question in another way. What proportion of offspring from this cross would you expect to be big Y question mark, big R question mark? In other words, this would be the same thing as saying yellow and round. To be yellow round. And so we'll do the same thing. We'll go down here. So this Punnett square is big Y, little y. Big Y is the cross, you know. Little y. Gonna get another piece of red. So this is big Y, big Y, big Y, little y, big Y, little y, little y, little y. So how many of these are actually going to be this? 3/4. That, and that, but not that. 3/4. And then the same thing would hold for this one. So this would be you won't even need to do these crosses in the future. I'm just doing them so you can clearly see. Again, it's these that were that, and it's these that are that. So this is 3/4. Multiply those together, and what do you get? 9/16. Ha ha! So let's do that. 9/16 is 9/16 is that 9 to the 9 of the 9 to 3 to 3 to 1. So this little kind of, what you can therefore do is break every chromosome's activities. Of course, there's a maternal and a paternal. It's a head and a tail. Every one of those you can break down into a smaller event, and then you can solve problems about specific outcomes, either presented as absolute specific outcomes with all the alleles, or kind of unknown outcomes, like a big Y question mark, big R question mark is how we did this one, which basically means how many of them were yellow and round. So this is a wonderful tool to help you solve complex problems. We'll actually do some quadra or penta hybrid crosses of all independent loci and use this exact principle to come up with some really cool answers and solve problems quickly, because there's no way, even when you get to a tri-hybrid cross of let's say tri-hybrid cross all independent loci, that would require, think about that. That's a question I want to ask you. How big does your Punnett square need to be there? You got to first figure out how many gametes that you get in a tri-hybrid, and then lay that thing out, and it gets big. I'll tell you right now, it's an 8 by 8. Again, you go back to the formula, x to the n. Okay, everybody. That could have lost you a little bit, and if it did, that's okay. I hope that it didn't. But the bottom line is, go right back to that fundamental cross that we did. We did a dihybrid cross. We started by making all the gametes. We lined them up equally, same direction, column, row. Ended up with, therefore, a 16-cell Punnett square. We filled it all out. We tallied everything up. We ended up with these genotypes in these proportions. And then and that was kind of meaningless in terms of a genotypic ratio. But then when we tallied the phenotype, we ended up with a really great story. The 9 to 3 to 3 to 1. That is the gold standard dihybrid cross phenotypic ratio, when at each locus it is normal complete dominance. So this is good stuff. All right, so that's all we have to do for setting up dihybrid crosses. Now let's take a look at some variations and modifications in dihybrid crosses, especially when genes interfere with each other. All right, this is that. It's turning it up, constantly getting cooler and cooler and cooler. So what we're going to talk about now is an interesting concept. We're going to still focus on. We're going to focus right now on two loci. So focus still. Whoops. Still on two loci that are independent. I'm going to call them two independent loci. Abbreviating independent, two independent loci. And now we're going to look at a phenomenon called epistasis. Very, very cool stuff. Epistasis is something where, defined as the expression, so the expression of one gene or gene pair masks, that's the deal, masks or modifies the expression of another gene or gene pair. There we go, epistasis. Now, that's a complicated definition. We really need to just look at examples for this to become clear. That's what we're going to do. We're going to look at examples of epistasis. It's very cool. What you need to do, though, is remember the base. We're all starting from, remember that we're starting from this kind of 9, 3, 3, 1. That's our basic kind of. This is a dihybrid cross. A dihybrid cross of two independent loci with complete dominance at each. at each. This is the expected phenotypic ratio. And what we're going to do in epistasis is that epistasis will alter this, and in specific ways. We're not going to see the 9-3 to 3-1 anymore. We're just going to start to see examples where it changes, and that's what I'm going to show you now, is epistasis. Okay. All right. So epistasis. Our first example is going to be a homozygous recessive masks another locus, masks the other locus. Let's take a look at the cross. So here's our example. You're going to get this process. This is a pigment system where precursor molecule, the precursor molecule is requires a big A question mark to become a black pigment molecule. And this would be a pigment molecule called melanin. And then it's the next locus that requires a dominant to get to what's called a Goody pattern. And this is kind of a salt and pepper look for the coat of mammal. So we're talking about mammal hair pigment, precursor molecule, and requires to get to the black phase with melanin requires at least a big A. It doesn't matter what the second one is; it has to have at least a big A. This is of course normal complete dominance, and then it has to have at least a big B to get to the Goody pattern. So here's how we look at this. Here's your F2s. And of course, this was a cross of this. We're always going to look at this cross. And by the way, these animals are a Goody for phenotype. And you can double check that here because each animal has a big A and a big B, so it gets all the way. One more time. Each animal in this cross has a big A and a big B, so it gets all the way from precursor to black to a Goody. So two agouti animals. This is, of course, the dihybrid cross. The dihybrid cross yields these F2s. We get, in this F2s, 9/16 of them are going to be big A, big B. What's the phenotype? You already know, agouti. 3/16 are going to be big A, question mark, little b, little b. It can't go there. These will be black. It doesn't have the ability to make it to a Goody because it needs a dominant B to get there, and it doesn't have one. 3/16, all these numbers came straight from our 4x4 kind of example of a dihybrid cross. We used Y and R. Now we're into A and B. So then 3/16 would be this. Now what happens here? They don't get to black. So they're basically going to be albino. And then 1/16 also doesn't get to black because it's little a, little b, little b. It's albino. So here is the magic moment. We therefore have our ratio for this kind of, this example. Our ratio produces a 9, whoops, got ahead of myself, a 9:3:4 dihybrid cross ratio. That is a classic ratio of this mechanism of epistasis, and it's one to memorize. And that's the whole process. Isn't that cool? Now we're looking at gene interactions, basically. That is exciting. Once you open it up and start to expand beyond one simple locus, yeah, you can have fun with one locus, especially if it's human blood typing or sickle cell anemia. But we really need to expand, and this locus impacts this locus, as you can clearly see. If you can't get to here, it doesn't matter what that is. Boom. All right, so that's our first. Let's do another example of epistasis. So our second example of epistasis, continuing, is that a dominant allele at one locus, either one, masks the expression of the other locus. So we got that. We'll look here at, our example will be fruit color in summer squash. Groceries. So fruit color in summer squash, let's just throw it up. 9/16, 3/16, 3/16, 1/16. So we'll do big A, big B. We'll get used to this. So here's what happens in our business. We're talking about a dominant allele. And it's going to be a dominant allele at the A locus. That and that make, it basically masks the expression of the other. So it really doesn't matter if it has a dominant allele, what's going on here. These guys end up being the same. So we end up with 12/16 being white for the squash. And then over here, we end up with 3/16 being green phenotype, because that's obviously what we're getting out of here. And then the homozygous recessive is 1/16 of a yellow squash. Of course, you can figure out how they could make these in the grocery store, right? Homozygous recessive, breed them on down the line. Go get yourself a yellow summer squash. So look at that. The white one is because of a dominant allele basically precludes, it wipes out anything that the B locus wants to say. That's another type of epistasis. So therefore, we must be prepared here to identify the 12 to 3:1 ratio of epistasis. That's great. So now we have 9:3:1, dihybrid cross independent loci, complete dominance. And then we have epistasis example number 1, and that's a, without naming it, that is a 9:3:4, and then now we have a 12:3:1 predicted ratio. Oh, that's exciting. I got one more, and here it goes. Okay. Number three is going to be at least one dominant allele at both loci. Wait a minute. Let me change this. Let me change this to a better definition. So, homozygous recessive at either locus masks a dominant at the other. At the other locus. So here's a pea flower color. Pea flower color. And 9/16 are this. 3/16 are this. And 3/16 are this. Whoops. And 1/16 is this. So here's the idea. Whenever you're going to have this homozygous recessive, which is shown here, which is shown here, of course, which is shown here double, but that doesn't have a dominant. So masks a dominant, masks a dominant. That basically means that you're going to end up, here's what it looks like. Pea flower color, you have a precursor substance, and that requires gene A, and then you have intermediate product that's been biochemically changed, and then you have big B that requires to the final pigment. So you can see that if you're recessive here, you can't go. If you're recessive here, you can't go. The only way to get colored is to go all the way. So this guy ends up being, this is the only way to get purple. 9/16 is purple. And then all of these together, 7/16 will end up being white. This is a 9:7 phenotypic ratio, and it is characteristic of this type of epistasis. So there you have it. Wow. So there's some other modifications. It's good to be familiar with ratios. It's good to be able to work through these kinds of things. I have a fabulous set of genetics problems. We're going to be working on those with some of my help in our Q&A session for next week, but you can certainly fire these up and get on them yourself. It's good work. It's fun. It's fun to solve problems. It's a change of pace. So I really hope that you get into it. I really hope that you do what's necessary. Do the job. Do what's necessary to totally understand how to do genetics problems, which means you totally understand the basics of inheritance and you know all the terminology that goes with it and how to use it. So that's what to do. And then, of course, the other jobs that are on your table, always, there's quizzes out there. We have a lot of people that have a lot of quizzes still to do. Definitely do the new quizzes, the ones that we're doing now with the chapters. But, man, why don't you just break out and do a quiz from the biochemistry chapter or something, you know, the macromolecule chapter. You want to get all these quizzes done. Of course, another job to do, exam 3. It's hot, it's ready. And of course, this is exam 4 material. So we have lots of things to do down the home stretch here. Stay positive, stay excited, and I'll see you next time.